All right, everybody. Thank you again for coming. This is Mr. Zawacki, and today we're going to be talking about the beginning of our biology EOC review, part 1, where today the scientific method, properties of water, and macromolecules, mostly from Module 1, beginning of Module 2. First thing, and one of the first things that you learn every year in every science course, is the scientific method. There are 6 steps, at least in this scientific method, that we need to know about, know the order, and know different parts of each step as well. And these 6 are right here. Step 1, you always ask a question. That starts everything. The question comes first. The topic or the question is always number 1 because that gets the whole scientific method started. Question number 2, you're going to want to do more observations and research. Look up the question. Get as much information as you can about the question. Step 3, make a hypothesis. We know what a hypothesis is. We've seen it for years. Hypothesis is an if, then, because statement. And it predicts something that's going to happen. It's your best educated guess on what the question is. Step 4, usually the biggest step of the 6 steps, is experimenting. With an experiment, usually will include variables. And this is something you guys absolutely need to know for the test. Independent variable is always what gets changed on purpose. So that's your independent variable. Dependent variable is data. Remember how we talked about that? Dependent is data, so that's what gets measured. On a graph, also, that's where you see the x-axis and y-axis. The x-axis is going to be the independent variable. So the pH of water in this certain example, well, they changed the pH from 8 to 7.5 to 7 to 6.5 to 6 to 5.5. They changed that on purpose. So that's the independent variable. That's the x-axis. Whereas the y-axis is the dependent variable, which in this case scenario, they're measuring the number of tadpoles or baby frogs. So that's another thing to keep in mind. Your y-axis is usually the dependent variable or data. The x-axis is the independent variable or what you change on purpose. Then step 5, you analyze the results. So once you get the results, you analyze them. That is the graph that you see below. So analyze, when they say analyze results, you want to think data tables, graphs, something visual that represents the data. And then step 6 is conclusion. We know what that is. That just summarizes everything that you did in the lab. So this is exactly what you guys need to know for the scientific method. You need to know the 6 steps: question, research, hypothesis, experiment, analyze, and conclusion. You also need to know independent and dependent variables in an experiment. Be able to pick them out. Independent variable is what you change on purpose. Dependent variable is what you measure. In this example here, they're measuring the effects of pH on tadpole survival. So the independent variable is the pH of water. That's the x-axis. Dependent variable, number of tadpoles, that's the y-axis. So that's something you also need to know. Okay, so that's it for the scientific method part. Know the 6 steps, know variables, know how to pick out the variables, and know how to set up the variables on a graph. If you can do that without reading from notes or looking up anything, you got it. You're good to go with the scientific method. Here's a sample question from you guys. You just want to type, well, actually, let me see if I can set up the polling here. Anyways, you want to read through this, and you're going to answer A, B, C, or D. So think about which one you think is the correct answer, and then I will put those in the poll up above, So all you have to do is select A, B, C, or D by clicking the little letter A above, right below my name. So select A, B, C, or D, or what you think the correct answer is. Give you guys about another 30 seconds because you're looking at the graph first, and then you're picking the answer that correctly describes it. And this kind of goes with your lab when you did in 2.02. So let's start talking about the data. It looks like we're struggling a little bit with this. We got some A's and some C's so far. So let's look. We're looking at osmosis in cells. That just means water moving in and out of cells. So the mass of eggs before soaking is 71, 98, 64. Average of mass of eggs after soaking, that means water went into vinegar, which is 95% water. Water went out of corn syrup, which is only 5% water. And then water went into distilled water. So, looks like that's kind of making sense. Then, based on the experiment, which of the following should be inferred about cells with semi-permeable membranes? Well, let's see which ones we can eliminate. Substances other than water may also cross the cell membrane. Not in terms of this actual data table, so that's not correct. Substances other than water may block pores in the cell membrane. That does not explain anything about this data table. Let's look at the other two. Water enters the cell when placed in environments of high water concentration, or water leaves the cell when placed in environments with a low concentration. So it's really now down to C or D. So think about which one makes the most sense in terms of the graph. Does water enter the cell when placed in environments with high water concentration? Or does water leave the cell? Well, here, look. Water is entering because it's getting heavier. Water is entering because it's getting heavier. So the answer is going to be C. Water enters the cell if they're in a high water concentration outside of the cell. That's just osmosis making everything equal. So that answer should be the letter C. So good job for those 5 of you that chose the letter C. All right, also learning more about characteristics of life. These are just things that all living things exhibit. So not just animals, but this is also anything that's living, down to a bacteria, really. Every single living thing has to have a cellular organization, which means they can either be unicellular, one cell, or they can be multicellular, many cells. Organization goes molecules, then organelles, then cells, tissues, organs, organ systems, organism. That's just the general organization of living things. Every living thing must be able to reproduce as well. So they're organized by cells, they must be able to reproduce. Asexual reproduction is one parent. It's a clone of a parent. Sexual reproduction is 2 parents. So they have genetic variation from 2 parents. All animals reproduce through sexual reproduction, Plants can do both. Other organisms could do both. But animals, it has to be two parents to create another one. With metabolism, every single thing must consume and be able to break down that food. Energy is required for all life processes. So, every single living thing is either an autotroph or a heterotroph. Autotrophs make their own food, so those are like plants, or bacteria sometimes. Heterotrophs eat other organisms, so that's all animals. All animals are heterotrophs. But all living things are one or the other. Also, all living things must know and try to make it to homeostasis. That just simply means being normal. Your body is always in a fight to make you normal, make you feel normal. And that's what homeostasis is. Every single living thing tries to make everything normal. So that's what homeostasis is about. Step 5, or the 5th characteristic, everything must be able to pass down the genetics. That kind of goes with reproduction, asexual or sexual reproduction. But when a new organism is made, the DNA must be passed down. So all living things pass down their DNA to the next generation. Every single living thing must be able to respond. All living things can at least respond to a stimulus. That means they can move or release a defense mechanism or change shape, whatever, but they have to be able to respond to stimulus. Every single living thing must be able to grow and develop too. Even one-cell bacteria still grows and develops. Obviously, animals do too. But every single living thing must be able to grow and develop throughout its lifetime. And the last thing, every single living thing evolves. Remember how we said that in Module 5? All living things change over time? Well, that's true. Every single living thing on Earth, from a bacteria to a human, will adapt and evolve for the better over time. And that's what we talked about in Module 5. So these are just characteristics of all life. Every single living thing must have cellular organization, must be able to reproduce, must be able to metabolize and break down things to make energy. Every single living thing must be in homeostasis to try to make the whole organism normal. Every single living thing must pass down DNA. Every single living thing must be able to respond to a stimulus. Every single living thing grows and develops. And every single living thing has the ability to evolve. So that's kind of the characteristics of all living things. Then, in 104, you got into the properties of water. Remember how water is way more important than we think it is. And with water, there's several important factors you need to know. First of all, it's a polar molecule. Second, cohesion-adhesion. Third, high specific heat. Fourth, its density is greatest at liquid temperature at 4 degrees Celsius. And then fifth, it's a universal solvent of life. So I'm going to explain what those mean here. First of all, with a polar molecule, that just means it has positive and negative ends. So with a polar molecule, you have H, and O. Water molecule kind of looks like Mickey Mouse ears. But here's your oxygen atom, here's your hydrogen, and here's another hydrogen. All polar means is that the hydrogens are positive, the oxygens are negative. That's all it means. But it acts like a magnet, and that's where you get cohesion and adhesion. Cohesion is where water molecules stick to their cells. So you have another O and an H and an H. Well, they stick to their cells. That's cohesion. Or water can stick to other things. That's called adhesion. Water isn't sticky, but it's polar, and that's what allows it to stick to other molecules. Also, you have a high specific heat. What that means is very simply, it takes a lot of energy to heat up and change the temperature of water because it has a high specific heat. Same thing with high heat of vaporization. It takes a lot of energy to heat up and boil water compared to other liquids. Also, the density is greatest at 4 degrees Celsius, which means if you have cold water, it is the most dense version of water. So if you put warm water with cold water, warm will go to the top. If you put an ice cube with cold water, the ice cube will still go to the top because water is most dense as a cold liquid. With any other form, it will be less dense. And last but not least, it's the universal solvent of life, which just means it dissolves lots of things. Remember how it's partially positive and partially negative? Well, because of that, it can dissolve things like salts and sugars and minerals, so it's the universal solvent of life as well. So definitely know what these words mean and how to explain them, just like I did, in your own words without reading from any notes. Know that it's a polar molecule. Cohesion is where it sticks to itself. Adhesion is where it sticks to something else. High specific heat just means it takes a lot of energy to heat up and change the temperature of water. Remember, water is most dense as a cold liquid, and it's the universal solvent of life, so it dissolves many different things. Here's the polarity of water again. Remember here how H2O looks like Mickey Mouse ears? Well, you got your hydrogen, and you got your oxygen, negative on the bottom, positive on the top. In a water molecule, 2 hydrogen atoms form a single polar covalent bond with an oxygen atom. This gives water more structure than other liquids. Because oxygen is electronegative, the region around oxygen is partially negative. The region near the 2 hydrogen atoms has partial positive. So remember, all you need to know about polarity, guys, is that the hydrogen end is positive and the oxygen end is negative. And the negative attracts other positive things, just like a magnet would. And the positive side attracts other negative things. That's why opposite ends attract. It happens on a magnet. It also happens in water molecules. All right. Couple questions for you guys before we get into the last couple slides. First of all, question number 1 for you guys to put in the bottom. Which property of water shows that water sticks to other substances? Cameron, perfect. Talea, good. Lauren, good. Good job, everybody else. It is adhesion. Remember, adhesion, it sticks to other things. Cohesion is where it sticks to itself. All right, so tell me 2 other additional properties of water besides adhesion. What are 2 other ones? We just went over 6 of them. Yep, universal solvent, cohesion. Anybody remember some other ones? Yep, it's a polar molecule, high specific heat. The density, remember, yeah, the solid is less dense than the liquid version. Very good. Third question, is ice more or less dense than water? Remember, put ice in water. Whatever floats to the top, that's less dense. So if you put ice inside of a glass of water, you will note that it floats to the top. Ice is less dense than water, for sure. And then what type of chemical bond does water have with itself? Just said that. What type of bond does water have with itself? Good job, Noah. Paying attention. Paris, good job. It is a covalent bond on how water binds with itself, or bonds with itself. There are ionic bonds when they bond with other things. But covalent is when it shares with itself. All right, so 2 more slides, or 3 more slides. This is the chemistry macromolecules. We're going to talk about it a little bit today, and then we're going to continue with this on the next live lesson. The biochemistry comes from 201 in your science biology course. And we should know that organisms are composed of organic compounds. All the word organic means contains carbon. That's all the word organic means. Macromolecules are the big molecules, such as carbohydrates, lipids, nucleic acids, and proteins. So these are the 4 main macromolecules, and they're made by dehydration synthesis. So with dehydration synthesis, does that mean adding water or taking away water? What do you think? Dehydration synthesis. That's what they're made by. Does that mean adding water or taking away water? Good job. Yep, everybody got that. Think of the word dehydration. So when you take away water, you are actually building macromolecules. When you add water, you're actually breaking them apart. So make sure you know that as well. Carbohydrates, their building block is monosaccharides. So the pieces of carbohydrates are called monosaccharides. The pieces of lipids are called fatty acids or triglycerides. The pieces of proteins are called amino acids. And the pieces of nucleic acids are called nucleotides. This is also very important. You need to know the building blocks or monomers of each macromolecule. Carbohydrates are made up of monosaccharides. Lipids are made up of fatty acids. Proteins are made up of amino acids. Nucleic acids are made up of nucleotides. Carbohydrates are your sugars and starches. Lipids are fats and oils for long-term energy storage. Nucleic acids, that's all your DNA and RNA, so that's all your genetic information. And proteins, well, they build muscle, they help protect the body. The enzyme is also an example of a protein, so they do many different things for the body. And then one last thing, you should know that carbohydrates and lipids are both made of carbon, hydrogen, oxygen. They're both made of those. Whereas proteins are carbon, hydrogen, oxygen, nitrogen, and nucleic acids are carbon, hydrogen, oxygen, nitrogen, and phosphorus. So all 4 are made up of carbon, hydrogen, and oxygen, but proteins add in a nitrogen, and nucleic acids add in a nitrogen and a phosphorus. So make sure you know the difference between those parts as well. All right, and then metabolism, remember we already said all living things must be able to contain and be able to break down stuff to make energy. All metabolism is controlled by the action of enzymes, which are proteins. We have enzymes in our bodies, and they help us out more than you would know. Without enzymes, our body would be working twice as hard to make energy. But because we have enzymes, they do all the work for us. Think of like you need to make a dinner tonight, you need to cook it yourself. Well, if you had an enzyme, it would prepare the whole meal for you. You just have to hit go. And that's exactly what enzymes do. They prepare reactions inside your body. So they help them speed up the reactions inside the cell. So they're proteins that speed up chemical reactions. They have a very specific shape and interact with a specific substrate. So here's your enzyme here, and it works like a perfect puzzle piece. And when that puzzle piece is active, it helps the chemical reaction take place. That's exactly what they do. They're reusable too. So when they're done, they go back to normal. But they have to work at very, very specific temperatures and pH. If the temperature gets thrown off in your body or the pH gets thrown off in your body, enzymes won't work. And you're going to have a really hard time making energy for yourself. That's why enzymes are very important, because they help reactions take place in your body. They prepare reactions for you, so you don't have to do it yourself. So that's why an enzyme, which is a protein, is a very important thing. All right, last question for you guys before we get into it, for the end of today. As food travels through the digestive system, it is exposed to a variety of pH levels. Stomach has a pH of 2, and the small intestine has a pH of 7 and 9. HCl, which is hydrochloric acid, converts pepsinogen into pepsin, an enzyme that digests proteins in the stomach. So the question is, which of the following most likely happens to the enzyme pepsin as it enters the small intestine? So as it goes from a pH of 2 to a pH of 7 to 9, what do you think is going to happen? So try for the poll, A, B, C, or D. Well, let's see. We have a majority of A, possibly because I might have already shown it. But remember, enzymes work at very specific temperatures and pH. When the temperature or pH changes drastically, they're not going to work anymore. So the stomach is a pH of 2, and the intestine is a pH of 7 or 8 or 9. So what happens when the enzyme goes from one to the other? Well, it becomes inactive, right? Because it won't work in both of those pHs. It'll work in one, but not the other one. It has nothing to do with replicating. It does not change the shape. It has only one specific shape. And the activity does not increase. It just becomes inactive. So make sure you guys know that the answer is A, and know why it's the letter A as well. It's because it becomes inactive after learning, and going through different pHs. And that's it. That is letter A. And that marks the end of today, guys, for what we learned about. Again, just going really quickly through everything to make sure we have a good understanding of what we need to know. If you guys can speak about these without looking at notes, just like having a conversation with somebody or with a parent, then you know you got it. You definitely need to know the 6 steps of the scientific method with examples of each step. You need to know independent variables and dependent variables. Remember, independent is what changes on purpose. Dependent is what gets measured as data. And you also need to know how to set it up in a graph. Independent variable is your x-axis. Dependent variable is your y-axis. You need to know also the characteristics of life. Every single living thing on Earth has to have cellular organization, be able to reproduce, be able to break down stuff to make energy, homeostasis being normal, passing down their DNA, heredity, being able to respond to a stimulus, being able to grow, develop, and being able to evolve and change. All living things have to do that. You should also know about the properties of water. Remember, polar just means positive and negative ends. Cohesion sticks to itself, adhesion sticks to other things. Know about the other ones as well, and be able to explain them. Also know about the macromolecules. You need to know the 4, carbohydrates, lipids, proteins, nucleic acids. But you also need to know what the four are made up of: monosaccharides, fatty acids, amino acids, nucleotides. And you should know examples of each and how they're different from each other. Absolutely. And you should also know that they're made by removing water, dehydration synthesis. And they're broken apart by adding water, which is called hydrolysis. And you should know enzymes. Enzymes help reactions inside our body. They do the reactions for us, so we don't have to. And they work at very, very specific pH and temperatures. And that's it for today, guys. If you're able to talk about those things and be able to understand what we just talked about, you're good. That's about 20% of your EOC exam, so we're on our way. So do you guys have any other questions for today?